So let's get started. We're going to go back and just quickly go through all of chapter 11 that we've discussed, just as a really quick review to get us up into speed and thinking about muscle again. Remember that our next exam is going to cover muscle, chapter 11, blood, which we're discussing now in lab this week, and the heart, which will be our topic for the next two labs, as we'll be looking at the cardiovascular system and the function of the heart. So what did we discuss last time? Remind me. Yeah, we talked about how muscle is a tissue, and we talked about the characteristics of that tissue, the characteristics of all muscle. Also went through the sarcomere units and the fact that skeletal muscle is striated. Now, the vast majority of this presentation is on skeletal muscle. Toward the end, there's probably about 15 slides on cardiac muscle and maybe 10 or so slides on smooth muscle. And at that point, I'll be asking you to compare and contrast the three muscle types in some more, some greater detail than what you've learned in the past. So we went through all the layers of the muscle. We talked about the different connective tissue layers that are associated with muscle. And we looked at what a muscle fiber is, what a muscle cell is, and some of the unique characteristics of that muscle cell. We then got into the conversation about the myofilaments, these contractile proteins, the myosin and the actin. Tell me, what is the thin filament? Let me quiz you on this. The thin filament? The thin filament is actin, but it's actually what? Two wounded F-actins, fibrous actin molecules. And what else is also associated on that thin filament? Troponin, which was a three-part protein, and also tropomyosin. Tropomyosin was like that rope that went around the entire actin, and troponin was what we've seen so far, the molecule that specifically binds what? What binds to the troponin? Calcium, right? So there was a portion of troponin called troponin C, and it was to that subunit that calcium was attached. And then remember that in the muscle, not only do you have actin and myosin, but you have some other regulatory proteins, and really the only one, I mentioned titin, but the one that has any clinical significance is that dystrophin molecule, which we recognize is mutated in different forms of muscular dystrophy. Look back at the A, the I, the Z, the H.
In a relaxed muscle.
And then that is a beautiful picture of what? That's a beautiful picture of a thin filament, right? So now we see the troponin, the little Matryoshka doll, the three little snowman. So there's your little troponin complex. And remember, there's an I, a C. There's a troponin I, which is inhibitory. We'll talk more about it. There's a troponin C to which calcium binds. And there's also a troponin. There's another part. Going blank right now, right? T, that binds to what? To the actual tropomyosin. Okay, so yeah, the troponin T is the part that binds to the tropomyosin. So we'll look at those a little bit more carefully. And boy, we know more about calcium now, don't we? So we recall that calcium is critical to the contraction of the muscle, that as long as there's calcium around, the muscle will continue to do the power strokes and the sliding filaments will continue to do their business. Where is that calcium stored? Calcium is stored in a specialized organelle called the sarcoplasmic reticulum. And the sarcoplasmic reticulum is wrapped around, it's this network of membranes that store calcium, and it's wrapped around all those, what are these tubular structures within the muscle cell? That is a what? That is a what? That is a what? Myofibril, right? So if you look inside a muscle cell, what you would see would be an abundance of myofibrils and quite a few mitochondria as well. Not a lot of cytoplasm, right? There really isn't much room for cytoplasm because we have to have all these proteins in there doing the contraction of the muscle. Around those myofibrils, there's going to be a network of membranes called the sarcoplasmic reticulum. And it's in a range, it's basically going around all those myofibrils. And what are these openings out here? These little openings, I call them little rabbit trails, right? Little rabbit holes. So what's going to zip down that rabbit hole? Yeah, the action potential, the electrical signal is going to travel along the membrane of the muscle cell. And what do we call that membrane? Sarcolemma. And that signal is going to dive down into the T-tubule. And that's what this opening is. That opening is for the T-tubule. And what does it do? It comes down here. And what are we going to find down in the T-tubule? On each side of the T-tubule, there's a triad. And on each side of the T-tubule, there's a cisternae, or a portion of the sarcoplasmic reticulum. And what is found on the surface of that sarcoplasmic reticulum? Calcium gates. What kind of calcium gates? What's going to open those gates? A voltage change or a molecule coming in? It's a voltage change, right? So these are going to be voltage-gated calcium channels that would open as that electrical signal travels down the T-tubule. That came, we already know, from the neuron that stimulated this muscle. Now the muscle, the neuron-muscle junction is called the what? Neuromuscular junction, right? Simple enough. And we mentioned that if there is no nerve going to a muscle, that the muscle wastes away. So there's a denervation effect, a denervation atrophy, as is on the previous slide. So if there is no nervous connection to a muscle, that muscle will undergo atrophy, which of course is a decrease in size and the number of cells within the muscle. A motor unit? Remind me, what's a motor unit? It's a neuron going down to a muscle, and more specifically, it's the neuron and all of the muscle fibers to which it innervates, right? So we're talking about, so some motor units are very small, going to very finely controlled muscles like of your fingers and your hands and of your eye, and other motor units are much larger, maybe as many as 1,000 fibers contracting when a signal comes down, and you can certainly see big motor units in a muscle like the gastrocnemius. The whole neuromuscular junction story is one we already know, right? Forget the fact that it's a neuron going to a muscle. It's really the same story as we learned in the nervous system when we talked about a neuron communicating with a neuron. It's still a synapse. There's still a release of neurotransmitters. There's still exocytosis of the products. It's actually more simple because here there's only one neurotransmitter we'll be discussing, and that is acetylcholine, which goes on to innervate all skeletal muscle. And what is on the other side of the cleft? Acetylcholine is released, and on the other side on the muscle would be what? Acetylcholine receptors. And what sort of receptors are they, even more specifically? Ligand-gated, so they receive the acetylcholine molecule, but when that channel opens, it allows what in? Sodium, right? So these are ligand-gated sodium channels. So you could say that the acetylcholine receptor is a what? Ligand-gated sodium channel. It's really describing the same thing. So when acetylcholine binds to the muscle, what happens? Sodium comes rushing in, and we have our lovely depolarization event and action potential, and we also call that an end plate potential, and that is just a specialized word for the action potential that is taken on by a muscle cell. That action potential is going to crawl along, I shouldn't say crawl, right? It's going awfully fast. It's going to move along the edge of the muscle cell, the sarcolemma. That signal is going to propagate down into those T-tubules, reach those voltage-gated calcium channels, open up the calcium, flood into the microfibrils and to the microfilaments, and now we know what happens. Right now, see, the calcium's going to bind to what? Calcium's going to bind to the troponin. And when the calcium binds to troponin C, what happens? Tropomyosin is moved out of its place, and as it's moved out of its place, it's actually uncovering what? The binding sites, so that myosin can now bind to actin. As long as calcium's there, the tropomyosin will stay out of the way. And as long as there's calcium, and as long as there's what else? ATP, right? Allowing this thing to be motorized, then we'll have the binding of the myosin to the actin. So what happens? Tropomyosin moves out of the way, myosin binds to actin, and there's already some energy stored in there from a previous ATP, and they bind and they do what? The head cocks, if you will. There's a power stroke. And as long as calcium is there and there's more ATP, what will happen? ATP will come in and do what? Hydrolyze or break that connection. You need ATP not only to contract but also to relax. So the myosin will be released by an ATP. The myosin will recock back, grab another actin as long as there's still calcium around and the tropomyosin's out of the way, and can repeat the thing, right? Repeat, repeat, repeat. Now, can it keep going forever? I mean, there's only so many, right? These filaments are sliding over each other. So there's a point at which you've come to the end of the rope. I oftentimes think of this as sort of being like, why am I going blank? Some sort of, okay, what did we play in middle school and high school? Tug-of-war, right? It's like a tug-of-war deal, thank you. And what would my arms be? Like I'm on the team and I'm winning, right? So what am I doing? I'm grabbing, that's my myosin, right? And yank on the rope, yank on the thin filament, on the actin. But at some point you get to the end of the rope, right? So we can't keep doing this forever, because at some point the sarcomeres would be as short and as overlapped as they could be. But that's what happens when you're maximally flexing a muscle, right? When you're maximally contracting a muscle, there's nothing more you can do, right? There's a maximum amount of work that a muscle can do because you are at the end of all the actins. So what makes a person stronger? I'm getting a little ahead of myself, but this is a story that I think we understand. If I want to build up my muscle mass, what am I going to do?
Constantly.
Right? I'm going to keep increasing the weights. I'm going to work out. I'm going to get more muscle mass. Now, more muscle mass is actually more muscle cells and more myofibrils. So the muscle cells actually get larger. There are more myofibrils, and therefore there's more actin and more myosin. So if I have more ropes to pull, I can do more contraction, more power, right? I can create more strength if I have more of these overlapping sarcomeres doing their job. Okay, so I got a little ahead of myself, but we've been through this, and I just want you to be seeing where we're heading. We've seen this beautiful picture of the neuromuscular junction. We know what's going on there. The only thing I didn't mention today is calcium is in this picture in two places, isn't it? It's not showing here, but calcium is going to be coming into the synaptic knob when the action potential comes down the neuron. That's a different population of calcium that will be necessary down here to activate the sliding filaments. Okay, so remember there's two buckets or two supplies of calcium, right? They're in two different places. They're different, but calcium's critical in both two places, right, for the contraction of muscle. So muscle is excitable, so was nervous tissue. The difference here is really the numbers. So the resting membrane potential of a muscle is at negative 90 millivolts, and the whole depolarization event's exactly the same. There's nothing different here, a different number, but the process is the same. Which gates open, the sodium, the potassium, the return back to the resting membrane potential is all the same. And then we got into some interesting scenarios. What sorts of things can interfere with neuromuscular junction function? So that would include some pesticides, some cholinesterase inhibitors, and the 3 or 4 that I mentioned included tetanus, right? So Clostridium tetani, an organism, a microorganism that's found in the soil, And this organism, we get a tetanus shot to protect us against this. But this organism produces a toxin, and that toxin does what? It basically overstimulates, right? What do you want to keep these things together? Tetany or tetanus toxin overstimulates the muscles. And so what will that do? Produces, so it's going to do what? Cause a tetanus, right? A constant contraction, overstimulates. Versus botulism, another toxin made by a different clostridial organism, and this one is going to cause, we know it's used, Botox, used for wrinkle removal, so it's going to cause the muscles to relax and not be able to contract. Why? What's the mechanism? What does it do? It blocks the release of acetylcholine, right? So if you don't have any neurotransmitter being released, then you cannot contract the muscle. And then I also mentioned this plant derivative curare, which was used by different groups in the past, and it competes for acetylcholine receptors. So again, the muscles cannot contract, and the animal or the person would drop unable to contract their muscles. So what are the steps in making muscle contract? Four steps of this process. Number one, you got to excite the muscle. So excitation. So that includes what? The action potential coming down, the release of acetylcholine, the picking up of the acetylcholine by the receptor, the acetylcholine, or not the acetylcholine, but the action potential traveling down the membrane to the T-tubules and going down to the sarcoplasmic reticulum. Then what happens? Then there's a, quote, coupling, right? A connection between this. So the coupling here is that as that action potential goes down the T-tubule and it reaches those voltage-gated calcium channels, the calcium will be released. And this is the, quote, coupling reaction. Now what happens to that calcium? It's going to bind to the troponin and cause the shifting of the tropomyosin and the binding of the actin and myosin, and now we're in the contraction phase of this whole process, and the need for ATP in the power stroke. And then relaxation. What do we have to get rid of? Name for me the things we have to get rid of to relax a muscle. We have to get rid of calcium, right? So how are we going to get rid of calcium? The sarcoplasmic reticulum is able to sequester that back very, very quickly through a protein called calsequestrin, right? Sequesters the calcium. And also there's an enzyme in the synaptic cleft called acetylcholine esterase, which is going to be able to very quickly destroy and break down any acetylcholine. So we have to get rid of the neurotransmitter, and we have to get rid of the calcium, because we recognize that as long as either one is around, when they're both around, we're going to have continued contraction. Well, ATP has to be there to relax, though, as well. So your cells are not going to get rid of the ATP. It has to be there. Until you die, you're going to have ATP around. Okay, so we're not worrying about getting rid of ATP in the immediate moment to relax, but we do need to get rid of the acetylcholine through acetylcholine esterase, and we do need to get rid of the calcium through the resequestering of the calcium back into the SR. Okay, but the ATP it can't go anywhere. It has to be there, ready to go. The body would never, another way of thinking about it from a teleological standpoint, but the body would never destroy ATP unneedingly, right? It's too expensive to make it, so we're not going to be breaking down ATP to solve the problem. That would be a very costly, wrong way to approach trying to fix something. Okay, so again, the relaxation requires the pumping back of the calcium and the destruction of the acetylcholine. So, any questions so far? Are we doing okay with this story? Pieces and parts. Look at your Connect assignments. It'll help you through this. Now, before the muscle begins to contract, there is an ideal sarcomere size, right? The sarcomere has the sliding filaments, the actins and myosins, and we recognize that if the sarcomeres within a muscle are overstretched or understretched, that it's not going to have the optimal ability to create the power. But there's an optimal distance, and that optimal resting length was between 2 and 2.25 micrometers. And we see that graphically shown here, that we have the maximum tension generated when we're at an optimum resting length of 2 to 2.25 microns. If we are overstretched or overly contracted or understretched, then we're not going to get as much power on each side of this curve. So this refers to tone. Your muscles are always being reset to their ideal distance so that when expected or required to contract, they're able to do so maximally. And that's all being done for you through your autonomic nervous system. And I'll be describing to you in a few moments some other mechanisms that keep your muscles at their ideal tension. That pretty much brings us to where we were, doesn't it? Okay? We pretty much got through all that last time, except for this as well. So we talked about a myogram, right? And in lab, what did we do? What did we measure our muscles with? Grip strength. And what was on the table, on the iWork screen, what did we see? We saw the curve go up, and we called that measurement a what? What kind of measurements were we doing? What were we reading? I should say, what was the readout? We were looking at an EMG, right? An electromyogram. And so here it's called a myogram, or in the case we had an electrical system, so an electromyogram. We'll do EKGs later, right? What's an EKG? Electrocardiogram, right? And there are EEGs, electroencephalograms. We've seen these terms, so I'm always surprised at how many people struggle with what is an EMG. Had lots of beautiful answers today. A lot of things about magnets. A lot of electromagnetic things. I'm not quite sure why, right? What's an EMG? Electromyogram, right? Don't make it harder than it is, right? So, now, when a muscle is activated through voluntary contraction, right? You send an action potential down from your brain, down a motor neuron. There's always going to be a little bit of a latency period, a little bit of a gap between the signal and the beginning of the contraction, and that is defined as being about a 2 millisecond delay. Makes sense? And then the muscle goes into its contractile phase, where the filaments are sliding over each other, and then we come back to the relaxation phase. So if I'm going to describe to you holistically what are the steps of your muscle, right? They're going to be activated, they're going to contract, and then they're going to relax. But at a molecular level, we had the 4 steps. At the 4 steps, we had the excitation, then the coupling of that excitation to contraction, then the contraction, and then the relaxation. So at a molecular level involving calcium and myofilaments, there's 4 steps, if you will, to muscle contraction. But if I'm going to say to you as a person what's going on with your overall muscles, we're just going to talk about, the initiation, the contraction, and then the relaxation. Okay, so this is about where we picked up. Neurons definitely have a all or none phenomenon. We know that that's true, that action potentials are all or none. They look exactly the same. That negative 70 up to a positive 35. That's what's going down a single neuron. In a muscle, there isn't quite this all or none phenomenon, because electrical stimulation follows all or none. So if a neuron comes down and innervates a muscle fiber, and it's strong enough of a signal, it will reach threshold, and every sarcomere within that muscle fiber will contract. Okay, but it's not true that the entire muscle fiber will follow an all or none, because there's a difference in intensity, and there's a difference in the amount of concentration of calcium, and there can be a lot of other differences that can vary a twitch. So what is a twitch? I skipped over that here. We did talk about it last time at the very end. A twitch would be the very quick or the minimum activation necessary to get a muscle to contract, right? It twitches. It's not very long. It's not sustained. It's not necessarily very strong, but clearly the muscle twitches. It moves quickly and restores back to its original place. But how long the muscle twitches and how strong the muscle twitches can change quite a bit. So let's talk about some of those different variations on this. Also, what else would influence the strength of a twitch? How stretched was the muscle before it was stimulated? Imagine for yourself, if your arm was straight out at 180 degrees and I stimulated this muscle, my biceps brachii, I have a lot of contraction that I have left to give, right? But if my arm is in this position, 90 degrees, and now I stimulate the muscle again, I don't have as much power, potential to do because the muscle was already in a partially contracted state. So we have a lot of variations here. It's not this all or none phenomenon like it was with a neuron. Also the temperature. We didn't do a temperature experiment. No one did that, but we know that our muscles, quote, warm up. We want to warm our muscles up to reduce injury, and then we want a, quote, cool down period. That's not only for your respiratory system, but also for the overall health of your muscles. So temperature also has an effect on your muscles. Here's a big one, pH. As your muscles are doing their work, what's being formed? If you're really asking a muscle to do its work, one of the byproducts of muscle metabolism is lactic acid and other muscles, or other metabolic acids. And as those acids accumulate, what's happening to the pH of the local environment in the muscle? It's going down. Well, low pH begins to cause fatigue of the muscles. So again, how likely are you going to be able to give a maximum contraction if the muscle has already been working and the pH has already been dropping and you're already reaching a state of fatigue? And then another characteristic is hydration. How hydrated are you? The amount of water definitely has an effect on muscle contractile force. Now those are all things that we didn't even talk about in that muscle physiology lab last week, right? I mean, you might now have some new ideas for your discussion or for your introduction if you wanted to grab a couple of those things, or if you wanted to, in your discussion of your paper, you know, what would I do next time? Well, maybe next time I do exactly the same experiment, but maybe I want next time to look at the effects of hydration on the patient. Somebody comes in dehydrated, somebody comes in hydrated. Or maybe next time somebody comes in and we do the same experiment outside in the cold, or we do it, you know, after warming up. So again, there's all sorts of variations on the theme. And last week we were all rather boring, right? We all did things at room temperature. We all did rather simple experiments, and that's okay. That's what you were asked to do. But you can see now that muscle contraction is a lot more than just an all or none phenomenon. So we need to be thinking about all those different situations. So contracting a muscle is a lot like a compact action potential. Now, I think that about 90% of the class finally understood the difference between an action potential and a compound action potential by the time we got to the lab exam. There were still a few, though, that were getting these concepts mixed up. An action potential is what? The signal going down a single neuron. It is all or none. It's always negative 70 up to positive 35, right? Never changes. A compound action potential was what? The cumulative activity seen by all of the axons traveling through a nerve simultaneously. And we saw that that compound action potential was graded. It could be a little bit of stimulation or more stimulation, and there was a maximum stimulation that the nerve could handle if all of the neurons were firing simultaneously. So a muscle's kind of the same way. It's going to be rare that every single muscle fiber in a muscle is being activated simultaneously. It's sort of like they're taking turns. So when you stimulate a nerve to higher and higher voltages, you saw higher and higher contractions. It's similar here, except who's sending that greater compound action potential? When you're going to contract muscle, you are voluntarily choosing, right? I know I need to lift up something heavy. So what am I going to do? I'm going to send a signal that says, hey, use more muscle to lift up something. If I know that what I'm about to lift is lighter, I'm going to, quote, recruit or send a lesser signal from my frontal lobe, right, from my motor strip, and I'm going to send it down my spinal cord and tell a fewer number of muscle fibers within the muscle to contract. I'm using the same muscle, right? I'm going to use my biceps brachii, and I'm going to lift a water bottle, and I'm going to lift a 100-pound weight, right? We get the difference. We all know that we can lift up a water bottle, and maybe some of us can pick up a 100-pound weight, and we're going to recruit a different number of fibers to do different jobs. Same idea as that compound action potential, except here we use the word recruitment, right? I'm recruiting more muscle fibers to do the job. Or we say MMUs, multiple motor units. Because what's a motor unit? One neuron and all of the muscle fibers that it innervates. So if I only need to lift a small weight, I'm only going to recruit or activate a limited number of neurons. Therefore, only a few muscle fibers will contract. But if I know I need to lift the heaviest thing I can, I'm going to do what? Send every signal, right? I'm going to send a massive signal, and I'm going to tell every single motor unit within that muscle to do the work. So we would say multiple motor unit summation, adding them all together, or this idea of recruitment. Does that make sense? Right? The common scenario. Now, what if, can you fool yourself? Have you ever been in a situation where you went to pick up a big bag and you assumed it was a bag of dirty laundry? So you put a little bit of heft into it and you realize someone's playing a trick on you, or maybe not, but it's all full of styrofoam peanuts. So you anticipated a greater weight than what it was. So when you went to pick that thing up, you had, quote, over-recruited, and when you lifted it, it went flying to the sky, right? Mm-hmm. Or return it around. You go to pick up a brick, and you think it's just a regular old clay brick, but it's a lead brick. So you under-recruited, and when you go to pick it up, it's like, oh, but what do you immediately do? Recalibrate, right? And you say, okay, I need to recruit more fibers to lift up the heavier brick, or I won't be tricked next time. I won't recruit as many fibers to pick up that bag of styrofoam peanuts. Right? So our everyday experiences, our common experiences, help us to fine-tune how many of these motor units we're going to recruit for each action that we do throughout the day. So what is this going to look like? In this graph, we've got the number of stimuli. So there's a stimulus, a little black line, right? See the little black line? That's a stimulus. That's a stimulus. If you look, there's a little bit of a delay before we reach the top. That little bit of delay time would be called the what? The latency, right? The latency would be a little bit of delay from the time that the stimulation occurs to the actual maximum contraction, or the beginning of the contraction. Even from the stimulus to the beginning, right? There's a little bit of a gap, 2 milliseconds or so. And then each of these humps would represent a twitch, right? The muscle has done something. That is assuming, in this picture, the stimuli are all the same. So all of the stimuli over here are equal, because we see an equal response. But what if I increase the stimulation, either in voltage? What if I gave it a greater voltage? What would I expect to see?
More.
Right, a higher peak, right? Because it would stimulate more muscles. But look what happens too. What if I am sending the same signal, but I'm doing it up to 10 times per second? If you're receiving, now this is amazing when you start thinking about this. If I'm sending an electrical signal 10 times per second, that's a lot of little signals, right? There's enough time for each stimulus to be produced and for a full recovery between the twitches. So your muscles can respond 10 times a second. Twitch and recover, twitch and recover, twitch and recover. It doesn't seem possible, but that's what the numbers are. You can have 10 stimuli per second and still get back to baseline. But what if I start stimulating a muscle somewhere between 10 and 20 times per second? I will then begin to see a staircase effect, what's called a Treppe phenomenon. And what's happening is that the muscle is still recovering between the twitches, but each time it starts to get a little bit more tension, and the SR doesn't have time to completely reabsorb all the calcium. So if I'm not reabsorbing all the calcium, then what's going to keep happening? Each twitch is going to get a little bit stronger. It's going to maintain a stronger amount of contraction. And also what's going on in here is that the more you're using a muscle, the more it's warming up. So as you warm up a muscle, also it'll become, the power of that muscle will increase. So the picture on the previous slide is of this staircase effect, right? It's the same stimulus. Each of these stimuli is equal, but they're coming at a rather rapid pace, and there isn't enough time. There's enough time for the muscle to relax, but there isn't enough time for all the calcium to get re-sequestered. So you start having this stair-step effect. It's a combination of the calcium and the temperature. Because the muscle's being used a lot, the temperature is going up. Now, what if you are twitching a muscle now 20 to 40 times per second? Now we get to what's called incomplete tetanus. I mean, each stimulus is coming before the previous twitch is even over, and now, notice back here, going back to the previous slide, see how they all come back to the bottom? They're all recovering back to the bottom, aren't they? The stair stepping is across the top, but they're all coming back to rest. But if I increase the intensity to the point of 20 to 40 stimuli per second, it's never coming back to the bottom. It is actually having a stair-step effect here because it's never able to relax completely. And there's also an increase on the top, a stair-step effect. Now that can go on for a while, but at some point, if we draw this arrow out for a while, we'll get to here. And at some point, if those stimuli are coming in so fast that there's no chance for the muscle to restore, relax, no chance for the calcium to get, to be sequestered, no chance for the acetylcholine to be wiped out of the cleft, now you will get to what's called a complete tetanus. And this is what tetanus does, right? Tetanus, Clostridium tetani, that toxin causes a what? Constant lockjaw or a constant contraction of the muscles, and there's no relaxation at all. So the word tetanus, right, we think about the shot we get every 10 years, but it's also the idea that a muscle is in full contraction and cannot relax. So if I'm going 20 to 40, I get incomplete tetanus. And again, one wave starts adding to the next wave, this idea of summation, right? So we have summation going on, and we get to a pretty much a state of tetanus, but not yet, not a complete tetanus. Complete tetanus will happen when you are at a 40 to 50 stimuli per second state. Can you even imagine, right? Electrical signals coming that fast. But that's what you're doing when you are going to lift something heavy, right? I'm going to lift something, I'm going to go to the gym and I'm going to lift my maximum weight. And I'm going to be doing what? I'm sending signals from my frontal lobe as fast as I possibly can. In fact, I'm telling the muscle to basically go into tetanus. I'm telling the muscle to contract at its maximum power. Now, how long will it last? Not very long, right? But I'm going to get the maximum power for a very short amount of time. So if you do a few twitches, right, more recovery, the muscle can keep on going longer before it gets fatigued. But if you get to a state of tetanus, there's absolutely no time for relaxation. The curve gets smooth, right? I mean, it actually gets to be a smooth curve. And honestly, in the body, we rarely get here. We rarely get over 25. So do we ever really get to tetanus in our body? No, because tetanus is not a good thing, right? We don't want to get to tetanus. So we really rarely get to a true tetanus. Now, I'll tell you this now, when we get to the cardiac muscle, we'll discuss this again, and we'll say the cardiac muscle never reaches tetany. And boy, is that ever a good thing, right? You would never want your heart to squeeze and stop, right? That's called death. So there are actually some safeguards into the cardiac muscle that we'll talk about. The good Lord put some safeguards in there to prevent tetany of the cardiac muscle. We'll discuss those differences as we get through today. So that's kind of a cool thing, right? This idea of stair step and the number of of contractions that you are receiving. Now, when we think about moving our muscles, we think about our body, right, contracting. But when someone gets electrocuted, what do they die of? Right? Heart failure. I mean, really, basically, right? You've got so much stimulation coming in and all your muscles, right? You ever seen, well, we don't want to watch somebody getting electrocuted, right? But what do you see? They go tense, right? Clearly, they go very, very tense. Their muscles are very, very tense, right? So that extra stimulation. So we send that stimulation through our nervous system, through our neurons to our muscles. We also could go to physical therapy and get electrodes put on us, right? And have our muscles stimulated. Have any of you had some sort of muscle stimulation, right? So again, what happens? Electrical stimulation is sent to you artificially through a machine, and you have no choice, but your muscles do what? They contract, right, in some sort of rhythmic way. So maybe you've experienced that. You know that electrical signal can be internal through your own nervous system or can be external through some device. And then there are situations where muscles contract unwantingly. Think epilepsy, right, where electrical signals are traveling from the brain down the spinal cord and people are spazzing uncontrollably, again, because a part of the brain is sending out signals without regulation. A couple of other terms about how muscles contract. Isometric versus isotonic contraction. Let's start with the word isometric. What does that word mean to you? Iso, same, metric, distance, right? A meter stick, measuring distance. So an isometric contraction is one where you're getting a lot of tension, but the muscle's not changing its length. Okay, everyone do this. Take your palms and push together. Push, push, push. Do you feel tension? Do you feel it? Are your muscles changing their length?
Yeah.
No, right? Your muscles are just sitting there. I'm not changing my length of my muscle. My muscles are staying in the same length, right? They're isometric. But if I push, I'll get to fatigue, won't I? I mean, I'll get tired from pushing on my own self or pushing on a wall. Just go push on a wall. Are your muscles changing their length? No, they're contracting, but they're not changing their length. That'd be an isometric situation, okay? And our muscles are doing this all the time, right? We're staying in one position, but our muscles are maintaining some tone. Versus isotonic. Iso, again, same. Here, you're changing length, but no change in tension. So isotonic contraction, there can be both concentric and eccentric. And concentric is what you're doing as you lift a weight, okay? So you lift a weight, you overcome the weight, the dumbbell, and clearly my biceps are changing their length as I contract. And so that would be isotonic, I'm overcoming the weight. Eccentric contraction would be, have you ever just picked up a dumbbell, really heavy, and just let it hang? Right? Now that's also doing some work for your muscles. So when you just pick up a dumbbell and let it hang, a heavy weight, you're still stressing your muscles and they're still getting a workout to a point, and that's where the muscle lengthens as it maintains the tension. There's a relationship, a graphical relationship between isometric and isotonic phases of contraction. So take a look at this graph. It'll make sense to you. But look at how these two things are the same. So for example, if I put a dumbbell in my right hand right now, right, and I'm not changing the length of my muscle, while it's in my hand, I'm not changing the length of my muscle, then for a while, that is isometric. But at some point, I'm going to overcome that weight, and I'm going to start changing my muscle's length, and now I'm moving into an isotonic situation. So look at that graph. It makes sense, that inverse relationship of these two types of contraction. Okay, questions up to that point? How are we doing? This is kind of fun stuff, isn't it? It's not, I know, it's not quite so esoteric, right? We all use our muscles. We may not think about our hormones, but we definitely use our muscles. How do our muscles get energy? So now we're going to talk about the different energy sources for muscle. Clearly we need ATP. We got to get it. But where do our muscles get the molecules to make the ATP? right? So what does one need to make ATP? Oxygen and a carbon source, right? A carbon source. You need to have, usually we think of sugars, right? Glucose. But if you didn't have any glucose, you could use some protein or you could use some fat. But you definitely need some fat or protein or sugar along with oxygen to do this whole thing called cellular respiration to make ATP. So we need to have an energy source, right? Usually glucose. It could be fatty acids. It could even be protein in an extreme situation. So if you've taken microbiology yet, you had a lovely, I think test number 2 goes into detail about glycolysis and about energy. And so I'm not going to revisit that in great depth. You'll get that at some point in your future, but let me just introduce the basics of that. That is, there's 2 ways to make ATP. The way that we prefer, if you will, in our body to make ATP is aerobic. Because in aerobic respiration, we've got plenty of oxygen. We get a lot of ATP out of it, and we don't get as many toxic end products. So this is what we live in most of the time, right? We live in an aerobic respiration state. Our body certainly is in this state. We're breathing oxygen. But at the microscopic level, this is where you're thinking about this. This is at the microscopic level, because clearly we're all aerobic beings. We don't have a choice of being anaerobic. We don't have a choice of living without oxygen. So I'm talking about at the microscopic level of the cells and of the tissues. Now, you can also make ATP during an anaerobic phase, but this is called fermentation. And during our muscles, what our muscles do is when they ferment, they make lactic acid. And when this anaerobic time, when there isn't enough oxygen immediately available to your muscles to keep up with the demands of your muscles, you are going to make lactic acid and you're not going to make very much ATP. So you really don't make very, very little ATP is being made. So your muscles want to stay in a state of aerobic respiration, but there will be times when you're working hard and exercising, you start feeling that lactic acid buildup. What that's telling you is that at the muscle level, even though you may be huffing and puffing and breathing in as much as you can, at that moment your muscles are not getting enough oxygen to keep up making the ATP they need. So we've got both an aerobic and an anaerobic option. Let's take a look at this. So we're going to be looking at the mode of ATP. Where are we getting this? And we're looking at this guy. And he's at the starting line, time 0. Now at this point, he's breathing normally. He's got regular old aerobic respiration. There's no lack of oxygen to his muscles. And there's a molecule down there that I've only mentioned in passing, myoglobin. Myoglobin was the hemoglobin-like molecule for muscle that kept adequate oxygen levels in the muscle so that when you begin to run, right, when you begin to use your muscles, you have oxygen available. Because when you begin to run, have you started huffing and puffing yet? Have your lungs begun to breathe more? Probably not, right? If right now the bear walks in the room and we all start running, we're going to have plenty of oxygen in our muscles at that moment from our myoglobin, but our respiration will take a little bit longer to catch up. So then, during the first few seconds, right, we're going to have our ATP because we've got oxygen available. But then, and these are rough numbers, within the first 10 seconds or so, we're going to switch over to a phosphagen system, and I'll describe that to you in a moment. And then a little bit later on, we're now going to be in a state of anaerobic. Even in those 20, 30 seconds, your muscles at the local area have run out of oxygen. And they're still having to make ATP. So they're now in a state of anaerobic respiration. They're going to start making that lactic acid. They're going to be less efficient in making ATP. And that will go and go and go until the person stops. Now, a well-trained athlete can stay in this state for a very, very long time, right? So you can work through this. You can become better trained and you can stay in a state of anaerobic respiration for a very, very long time. This is why runners are going to carbo load the day before. They're going to make sure there's plenty of sugars around that will be broken down slowly and make available to their metabolism. And then when you're all done running, Mary, that's a marathon. For me, that's across the parking lot. Now what are we going to do? We're now going to repay the oxygen, because what are we going to do after we stop running? We're going to keep huffing and puffing, and what we're really doing is restoring that oxygen back to baseline. Well, for Mary, that only takes a couple seconds for her body to come back to normal because she's so efficient as a runner. But for me, I'm going to be huffing and puffing for a while, right? Because my body's not used to it, and I've got a lot of restoring to do to get me back to baseline. So there's always this thing called repayment or oxygen debt that comes as we finish exercising, no matter what it is, right? We'll continue to huff and puff after we stop exercising. So, and once we get back to that, now we go back to aerobic respiration. We're back to normal resting metabolism. Vince?
So the breathing that goes on while you're actually running, that's not sufficient for all your muscles?
It depends on the extent and the amount of work you're having to do, right? And certainly with training. So a well-tuned athlete can go a lot longer, and they've got more muscle and they've got more myoglobin. They're ready to do more work, and they can keep on going. Other people, I mean, this is the variation, right? I mean, this could be minutes, seconds, minutes to hours, based upon your fitness level and your muscle training. I mean, we all can start, and we can all get to some point before we start huffing and puffing at the end. But it's this gap right here in the dark line what really defines an athlete from a non-athlete. How long you can go in this anaerobic state. Okay. A little bit more. There's a few details in here that I want to point out to you. And I know it's hard to see this. So this is percentage of energy expenditure, 0 to 30 minutes. And then this is with mild exercise, moderate exercise, and heavy exercise. Okay. So that's what we're seeing on this slide. Now, percentage of energy. So now over here we have glucose. It says plasma glucose. That's the glucose that's already in your blood, that's already part of you. Then, after that, once your glucose is gone, and it doesn't take very long for your glucose to get used up, then we go to fatty acids. And you've got fatty acids in your blood as well. And then after that, we start looking at muscles, triglycerides, and eventually your muscle also has stored in it some glycogen. And glycogen is a polymer of glucose that is specifically stored in your muscle, made by the liver in times of need. And so what you can see is under mild exercise, we'll use up the glucose in the plasma, and we'll use up most of the plasma fatty acids, and we barely get into, within 30 minutes, we don't get into much of the muscle triglycerides. If you're doing moderate exercise, okay, we're going to burn through the plasma and the triglycerides the fatty acids pretty quick. We're going to burn through the muscle triglycerides, and we're going to start diving into, right, the amount of muscle glycogen that's around. And if you're doing heavier exercise, you know, you're going to be using up a significant amount of your muscle glycogen. It makes sense. That's just the order that your body's going to grab the energy. First the glucose in your blood, then the fatty acids in your blood, then the muscle triglycerides, and then going on to the muscle glycogen. Now, in long term, right, percentage of energy expenditure during the later minutes of exercise, I'm not going to have you worry about that. I just want you to appreciate this graph, sort of the differences there. Okay. Now, also look right here. This is glucose, leg glucose uptake. So you're on a cycle of some sort. And if you ever use like an exercise bicycle, it'll have watts on there sometimes, the amount of work that you're doing. And so this would be a low, a medium, and a high resistance and a high number of watts that you are generating. And again, look at the colors. So you're going to be using more glucose. So if you're not doing much work, you're not going to take up as much glucose. If you're doing more work, then you'll take up more glucose. That makes sense, because your muscles need that energy. They need the ATP. We understand that muscles need ATP, right? They can't do their job. So they've got to keep making ATP. How do they make it? Glucose first, triglycerides second, or fatty acid second, muscle, triglycerides, and then the glycogen third or fourth. So it's all here in words for us. So in a short intense exercise, like a 100-meter dash. That's long for me, but anyway, a short, intense exercise, right? 100-meter dash. You're pretty much going to get all the oxygen you need from the myoglobin, right? It's there. It's sitting there. That's a relatively short distance. It's very rapidly depleted, but you're going to be done before you are done with it. You might need to dip into a little bit of this phosphagen group. In other words, muscles might look at some of this phosphate groups also to make the ATP. Now, when the muscles needs to get phosphate transfers, I said the phosphagen system a moment ago. When the muscle needs to start doing this, there are two enzymes that are going to start making ATP. The enzymes are myokinase. Now, whenever you see the word ACE, or the ending ACE, you know it's an enzyme. A kinase is an enzyme that adds adds a phosphate. So it's a kinase found only in muscle. What's the problem here? Let's make sense of this. This is a lot of new words, but the common sense is this. The muscle needs ATP. It can't keep up with the ATP demands. It used up all the glucose that was immediately available. It used up all the oxygen that was immediately available, but the muscle is still being asked to do work. It needs more ATP. Where's it going to get it? This enzyme, myokinase, can take an ADP, remember that's a molecule that's already kind of spent its 1-phosphate, and it can convert it back to ATP. So this enzyme can do what? Make more ATP, but without the oxygen need. There's another enzyme, creatine kinase. Now, creatine kinase can take a phosphate from a molecule called creatine phosphate. Anybody here take creatine phosphate as a supplement? Right? Some athletes will take creatine phosphate. It is basically a molecule you can put in your milkshake, in your smoothie, in your daily exercise, in your routine. And what you're basically doing is giving your muscles an extra molecule to make ATP from. So it's the kind of molecule you take for about a week. You kind of wean yourself up to it to a maximum amount. Then while you're in a training regimen or during the season of a football season, this is going to be something that short-term athletes use. This is not a marathon running kind of supplement, but this is something a football player, a sprinter would use. And they're going to keep this creatine phosphate levels in their blood pretty high or in their muscles pretty high, so that when they're in that short spurt, their body has another place to get more ATP in the short term. Okay, and that's part of this phosphagen system. Very, very fast. So here's that term again, right? We're going to get more ATP from what's there. So this is all it takes. In 1 minute of brisk walking, you've used up all the glucose in your muscles. You need to start going to the phosphagen system. In just 6 seconds of sprinting, right? So that's your average football player, right? I mean, a 6-second spurt for a play, and then, unless it's a big play, they're done, right? They run for 6 seconds, and then they recover, and they run for 6 seconds, and they recover. This isn't a basketball player, right? They're moving. But a baseball player, a football player, where they have those, or a short-term runner, short track person, would definitely benefit from having this creatine phosphate available to them. And or a weightlifter, right? Very quick spurts of activity and then rest in between. So this is where the phosphagen system is really, really important. And here's just what these enzymes do. Again, here's the myokinase. What's it doing? It's taking an ADP and making a ATP. Likewise, the creatine kinase is taking this creatine phosphate and quickly making creatine, and then the creatine can help to make more ATP as well. So again, two different ways of getting some more immediate energy for the muscles. It's in our body anyway, and we can supplement it by going to GNC and buying some creatine phosphate, or creatine phosphate. So as soon as we go through all the phosphagen system, which also doesn't take very long, now you're shifting to an anaerobic fermentation state. So we go through the glucose very, very quickly. Then we go to the phosphagens, and we use up all that's available there. And then the muscle finds itself, I still need ATP, but I'm not getting enough oxygen. So it goes to an anaerobic state. The problem is here, right? You're not getting much ATP. You're only getting 2 ATPs for every glucose. when you have plenty of oxygen, you're getting about 36. So there's an 18-fold difference here. Okay, so during aerobic respiration, you're getting about 36 ATPs for every glucose. During anaerobic, only 2. And the other problem is, in anaerobic, you're building up lactic acid. And that lactic acid is accumulating in your muscles and is dropping the pH. And if you're a highly tuned, athlete, you can work through that muscle burn and go further and further. But for a lot of untrained people, it doesn't take a whole lot of that muscle burn to say, whoa, I'm done, right? I'm stopping. Where do we get this from? Again, there's glycogen. There's what's called the glycogen lactic acid system. And this is going to convert some of that energy in your muscles, that glycogen, to lactic acid. Again, this is only enough ATP for another 30 to 40 seconds of maximum activity. Now notice I said maximum, right? So if you're going all out, you're going to go through the glucose quickly, you're going to burn through the phosphagen quickly, now you're going to burn through the anaerobic within 30 to 40 seconds, and that would be it, right? Now what happens? By the time you have been exercising for 30 to 40 seconds, what now has occurred? Your muscles are warmer, but what else has happened? You're breathing heavier. Your cardiovascular system, you've increased your heart rate, you've increased your respiration rate, and as a result, now you can play catch up a little bit. Not completely, not like the huffing and puffing at the end of exercise, but now for you to continue to run or to continue to do exercise, your cardiovascular system has caught up. Now you're getting more oxygen to your tissues, and now we can go back and hopefully keep up with an aerobic state in the muscles. And again, this can go for minutes to, however long as your body is trained to do this, right? So you have to have good cardiovascular function to get that oxygen back to recover. And then at some point, right? And now during that aerobic, when you come back to aerobic, you're back to being able to get 36 ATPs per glucose. Okay? We just keep on going and going and going. And when you're back to aerobic, there's not much lactic acid accumulating. So if you can get through that first lactic acid crisis, you can usually be free of lactic acid for a while. But even though your body is still giving you plenty of oxygen, now where are you getting the glucose? Where are you getting the carbon? So we burn through that glucose quickly. We burn through the fatty acids. And after a while, you're going to start taking, right, carbon sources from your glycogen and other sources. So what does this all lead to? Right? Whatever your state of conditioning is, you're going to lead to a point of fatigue, a progressive weakness, a loss of the ability of the muscles to contract. It can happen quickly if it's a skeletal muscle, right? Squeeze a ball, carry in. Why do your arms give out so quickly when you're carrying groceries? Does that ever happen to you? Right? Grab 3 or 4 bags of groceries and you're carrying them in your arms at like 90 degrees? What are you doing? Constant contraction. You're back to what? When you're carrying groceries in the crook of your elbow and you got 3 or 4 bags in your elbow and you carry them in, your arm's like, oh, it hurts, right? My arm's worn out. What kind of contraction is that? Isometric, right? Just like pushing against yourself or pushing against a wall. Clearly you're doing work, right? It's contracting the muscle and it can exhaust rather quickly. So muscle reaches that state of fatigue. Now, who mentioned fatigue yesterday or Tuesday? Someone said, what is fatigue? The question came up in some context. And I said, fatigue is many things. So let's look at some of the different things that fatigue, what can contribute to fatigue. Number one, you've gone through all of your glycogen. Okay, so the glycogen is the stored sugar molecule in your muscles. It could be, now think about this, if I have a shortage of ATP, right, I no longer have a sugar source, I've gone through all my glycogen, and I'm no longer making ATP, we've talked about the sodium-potassium pump. And you know that the sodium-potassium pump is a beautiful example of active transport, right? Which means what? It requires energy. It requires ATP. Now, what does that sodium-potassium pump maintain? Resting membrane potential. If you can't maintain your resting membrane potential of a neuron, and we talked about how you could add a chlorine or something else and make it more difficult to reach threshold, same idea. If you're mucking up your sodium-potassium pump and you're no longer able to maintain your resting membrane potential, it can make it more difficult to reach threshold. I didn't give you a number for threshold, right? I didn't say the number the threshold is, but clearly if you're lowering your resting membrane potential, it's going to make it more difficult for the muscle to get to that threshold point. Also, that lactic acid, that one we've talked about, lactic acid starts to build up. What does low pH do? What happens to a protein in high temperature or low pH? It dissociates or it denatures, right? So if you've got proteins that are in a lower pH environment, they're not going to work well. So as that lactic acid builds up and as the pH is dropping, you're going to start to inhibit, destroy enzymes that are necessary for contraction, that are necessary for more ATP synthesis. We're not done, right? Also, as you are continuing to contract your muscles, there's going to be, over time, an accumulation of extracellular potassium. Now, again, where does potassium usually find itself? Inside, right? But if you're asking your muscles to contract constantly and that sodium-potassium thing's going back and forth all the time, you're going to start getting more potassium outside. Again, what's that going to do? Hyperpolarize, right? You lose potassium. The inside of the cell is now what? More. Here's my cell. I'm inside my cell. If I'm losing potassium out, then my cell is becoming more negative, right? It's lower in its resting membrane potential, and that would make it more difficult, once again, to do what? Reach threshold. More difficult to cause contraction. Also, it's possible that your motor nerve, right, your neurons sending the signals have actually gone through all their acetylcholine, right? At the extreme case, you've run out of acetylcholine, you've run out of neurotransmitter, there's nothing else to activate the muscle. So, oh my goodness, I mean, there's all sorts of things, right? And when we run out of acetylcholine, we would call that junctional fatigue, right? It's not about potassium, it's not about sodium, it's all just about at the junction, we've run out of neurotransmitter, we can't contract anymore. So all of those things are going to contribute to fatigue. Thoughts on fatigue? Not just one thing, is there? Quite a few things, and they can all contribute collectively as well. Anything else? Yeah, Vince.
That's kind of a random question, but with the glucose that's used up first and in a short period of time, what does the body do to preserve that for specifically the brain?
Good point, because the brain, good grab. Remember that the brain needs glucose, right? What can exercise do to you?
We can use it up.
Make you a little dizzy?
Yeah.
Make you a little faint? Okay, what's going on? The body is switching from glucose is available. You've just used up most of the glucose through energy, expended through exercise, and you feel a little bit foggy, a little bit like you might faint a little bit, and the brain is having to wait for that switch. The body can make other molecules, right? Take other molecules to make the glucose for the brain. It has to. It absolutely has to. So, but that's exactly what's going on. Good point. I don't have a long answer for you. But clearly when you're using up that glucose, you can feel a little faint, and it's because the brain's not at that moment getting the maximum glucose that it needs. Darlene?
So then when you're exercising and you're being really vigorous, is it better to drink a sports drink than that has, or water?
It depends on how much you're sweating too. I mean, you want to stay hydrated. You want your muscles to be hydrated. If you're sweating a lot, you definitely want to be replacing it with some electrolytes. I mean, you are sweating out some electrolytes, and you want to bring those back into balance, but you can also overdo that. you can overdo electrolytes. So again, typically anyone who tries something new and vigorous will find themselves, they'll find what works for them. And you can read about what's ideal for the kind of exercise that you're doing. But, how many supplements, what kind of hydration, how often you should be taking in electrolytes, all that thing is, watch any kind of sports show and they'll kind of give you some suggestions for those things. Any other questions about fatigue or, well, how do we work against fatigue? We build up our endurance, right? So let's talk about what that really means. So if you're able to maintain a high intensity for more than 4 or 5 minutes, then you are certainly increasing your endurance. Now, a large part of your ability to endure is going to be based upon your VO2 max. This is your maximum oxygen uptake. Okay, because what did I say? The ability for you to keep on going, right? We all burn up the glucose, we all go through the fatty acids, we all go through the phosphagen system, but then what was it that allowed you to keep on going? Your body returned to aerobic respiration because there was an increase in respiratory function. So the ability for you to keep on going through a longer exercise protocol is going to depend upon how much oxygen you are able to uptake. So this is going to be the key. So your maximum oxygen uptake, the point at which, right, your oxygen consumption reaches a plateau and does not increase, and this is going to be proportional to your body size. When we do the respiratory system, second or third lab from the end, you'll all be measuring your normal lung capacity. And what you'll see there is that as you age, the expected lung capacity goes down, and that your lung capacity is maximum at around age 20. Also, the taller you are, the bigger your lungs are. And so the taller you are and the younger you are, the more you would expect your maximum capacity to be, your capacity to be larger. And as you age, or shorter people would have less capacity of their lungs. Again, normally, typically greater in males, and this can be hugely different in a trained endurance athlete. I don't remember the numbers offhand, but I remember Lance Armstrong, they used to say that his VO2 max was just amazing. It was off the chart, like freakishly off the chart. So not only, he dipped into a few illegal things, but he was just a massive animal on top of it, right? So pros and cons there. I won't go into the ethics of all that. But clearly he was a monster as far as his VO2 max and his lung capacity and his ability to make up time whenever he was going up, right? He made up time going up the hill. When everyone else was suffering. So he had an amazing lung capacity. So again, what can you do? If you want to beat fatigue in the short term, right, again, you could increase your ATP availability by taking some creatine, right, go down to GNC, and that's going to be really good for those weightlifting, short spurt kinds of activities. No risks that I'm aware of, except be careful if you have any kind of kidney disease, because this molecule does have to be cleared through your kidneys, and you don't want to overload an already damaged or difficult kidney. So you definitely want to ramp up on this stuff slowly and be really, really careful and work through a doctor's recommendation as far as checking your kidney function. Kidneys are already potentially at risk for damage, yeah. So you don't want to push it. Absolutely. Yeah. Definitely stay hydrated, right. So again, you want to stay really well hydrated so your kidneys can clear this stuff as well. Now, another thing you're going to do to increase your endurance is carbo load. Now, this is going to be important, especially for a longer endurance athlete. Usually the day before, right, they're going to go and have a pasta dinner or something before a big race, or keep and they're going to be eating complex carbs. You don't want to be eating simple carbs, simple sugars. Those get burned up and used by the body very quickly and kind of rise and crash. But complex carbs are ones that take our body longer to break down. So you may have had complex carbs last night, but over the night and through the next day, your body's still breaking down those complex carbs and releasing simple sugars, giving your body another source of constant sugar supply. So the bottom statement down here is that, that even though you're carbo loading and even though you're taking in a fair amount of food, apparently the energy availability is more of an advantage than the being, quote, weighted down by the food. So I don't have any personal experience with that. So while you're breathing, or sorry, while you're exercising vigorously, obviously you've got heavier breathing going on. And then when you finally stop, right, you're going to have post-exercise oxygen consumption when you finish. And the reason that is you're replacing the oxygen that has been lost during the first minutes of exercise. Because, think about it, it was during the first minute that you used up the oxygen, right? And then while you were maintaining your activity, you were back to aerobic. You were getting adequate oxygen. If you weren't getting adequate oxygen, can you keep going?
No.
No, right? You cannot stay in a state of anaerobic fermentation. So if your body is not able to kick out of that anaerobic state and get back to enough oxygen and enough cardiovascular and respiratory function to get back to respiration normal, you're not going to be able to continue exercising. Which is why a person with any kind of lung dysfunction, a person with emphysema, no matter what it is, or asthma, they're not going to be able to exercise as long or hard, no matter how much they want to, because their body will not be able to get enough oxygen to their muscles to maintain that level of activity. During exercise, well, I should say at rest, the amount of oxygen you're taking in is minimal, but you need a lot of extra oxygen during strenuous exercise. So the numbers just go really, really, really high. So when you're doing your post-exercise oxygen, you're huffing and puffing at the end, really all you're doing is catching up for the oxygen that you lost during that first minute. Right? Because we assume from minute 2 to wherever you stopped activity, your body was back into an aerobic state. So that minute afterwards really reflects the debt that you need to replace from the first minute when your body wasn't yet hyped up. You also need to replace the phosphagen system molecules, and that extra oxygen that you're taking in is helping to what we call oxidize the lactic acid. That is basically get rid of it. Okay, so that lactic acid that built up during that anaerobic phase, by taking in extra oxygen at the end, you're oxidizing it, and that is you are getting rid of it. Now there's something else that happens. What would help you? What would help a runner not have all the negative effects just before they start to race? I'm assuming that we're starting at ground zero, right? I just stand at the starting line and start running. What would an athlete do before they started to run? Stretch, get the muscles warmer, breathe, right? Start that breathing routine so that they do have more oxygen available to them at the beginning of the race. That's going to help them get through that first minute and continue at a sustained rate much, much better, right? And that was the big deal with the horse, with the nasal strips, right? Was it an unfair advantage for the horse last month to have nasal strips? You guys following the Kentucky Derby? Right? The horse that won the first two, they put nasal strips just like you do when you have a cold, those just little flaring nasal strips. And there was a big stink that they're giving the horse an unfair advantage by opening up his nasal passages. It wasn't in the rules, so they determined that it wasn't wrong. And then the second race, the jockey wore it. So the jockey's wearing a nasal strip and the horse is wearing a nasal strip. It was a tag team, right? And I'm sure the sale of those things went through the roof as a result. And then, anyway, it was a big deal. And then he lost the third race anyway, so it didn't matter. But, yeah, we all need those nasal strips, right? All they are is just a little bit of elastic fabric, right? They just keep your nasal passages open. There's no magic in them. But anyway, that was a big deal in the horse world last month.
I didn't know they weren't.
Well, they do. If your nose is stuffed, you know, I guess there's a little bit more airflow potential. I don't know how effective they are. Okay. So we realize we need to get our muscles, we had to repay the oxygen debt back to our muscles. Okay, stop there for a second. Any questions so far? Okay. Well, now we're going to talk about different types of muscle fibers. it turns out that we all know about it. Chicken, right? There's white meat and dark meat, dark meat. So we know that, in a chicken and a bird, that these muscle fibers are already sort of distributed, and we already understand this. There's white meat and dark meat. Same is true in your muscles. You have what's called slow oxidative, SO. They're also called the slow twitch, or the red, or the type 1 fibers. I wish it wasn't this way. There's lots of names to describe these slow oxidative fibers. Now, these slow oxidative fibers are huge in mitochondria. They have a lot of myoglobin, and they are highly vascularized. This kind of muscle is adapted for aerobic respiration and is resistant to fatigue. So what muscles in your body are more resistant to fatigue? Well, hold on. The soleus, okay, we'll talk about this in a second, but the soleus and your postural muscles, right, clearly our erector spinae muscles, the ones that keep our posture up, I mean, they don't tend to fatigue. We don't think about them very much. They're kind of working all day long as we're erect. So some muscles in our body are less prone to resistance because they have more of these slow oxidative or red type 1 fibers. There's also, though, fast glycolytic fibers. They're called the fast twitch, or the white fibers, or the type 2 fibers. These are much more about quick responses, but they fatigue quite easily. They're not fatigue resistant. These are the muscles that fatigue rather quickly. They are very, very rich in enzymes of the phosphagen system, and they also are really, really good at making lactic acid, which we know now can contribute to fatigue. They do not have a lot of mitochondria. They do not have a lot of oxygen. They do not have as much blood flow. That's why they are white, right? So the red fibers were more vascular, more oxygen, more blood. These white fibers are more about, they're really better for what? Anaerobic, right? They're really meant to generate lactic acid, but they also fatigue rather quickly. So, fortunate or unfortunate, our biceps brachii is largely fast glycolytic. We use it quickly, but our biceps brachii is pretty resistant to fatigue when compared to other muscles in the body, and the gastrocnemius actually fits here. So the gastrocnemius is more fatigue potential, but the soleus is more of a long-term muscle, okay, that's resistant to fatigue. So if you were to take a look at your muscle and stain it, right, to add some specific stains, then you would see an arrangement like this. You would see these are actual muscle fibers, muscle cells. So maybe this is part of a fascicle and this is part of a fascicle. And if you were to stain for something like myoglobin, right, what did I just say? Myoglobin was found where? In the type 1 in the slow oxidatives, right? So if I were to stain these cells for the presence of myoglobin, I would see more myoglobin in all of the slower oxidative cells. And then the pink ones would be the fast glycolytic cells. So you have a natural arrangement or distribution of these fibers in your body. Again, the soleus is mostly in endurance. This is your walking muscle. It just keeps on going and going and going. But your gastrocnemius is more your jumping jack muscle. It's more for jumping, so that's more fatigue, potential for fatigue. So your gastrocnemius mostly fast glycolytic, your soleus mostly, so easy, right? Soleus, slow oxidative, SO, you'll never get it wrong, right? So the soleus is much more a slow oxidative type fiber. So what do we have in the chicken? White and dark meat. Where is the white meat? The chest, the breast. Why? It better be resistant to fatigue. These suckers are flying. Well, birds in general. Okay? The white and dark meat distribution of chickens is the same in all birds. Okay? So, feathers, right? Resistant to fatigue versus legs. Fatigue, right? Darker. Can we change this? I mean, largely the distribution of muscle fibers is largely genetic. If you're a sprinter, thank your parents and your grandparents, right? It's as much about genetics as it is your training. You can train, I mean, anybody can train to be a sprinter, but only those individuals who have been blessed with the right kind of fibers in their genetics are truly going to be the outstanding sprinter, okay? So if you look across, and I don't want to say anything that's going to be taken the wrong way, but you look across the athletic world and you see certain ethnic groups and certain individuals who are simply genetically able to do certain kinds of athletic activities better than others. There are always the exceptions, right? So I don't want to say anything that's going to be taken as racist or anything else, but certainly some groups of individuals genetically have different combinations of fibers that make them long-distance individuals versus short-distance athletes. Just watch the Olympics. Okay. Any questions there? So we can change our white and glycolytic fibers a little bit by training, but largely we're born with it, and what we've got is what we're born with. And if you want to be a sprinter and you don't have the right genes, then go be something else, right? There's a sport for everybody. Part of it's your genetics, part of it's your training, part of it is your interest.
Go dance.
Yeah, go dance. Be a square dancer, right?
Archery.
Yeah, archery. I was on the bowling team. Varsity. Okay, so yeah, there's a sport for everybody, right? Okay, don't laugh, Vince. Okay, so I'm proud of my letter. Okay, so my son says that's not a sport. Okay. So, any questions so far? Let's take a quick break here, and we'll move on through some more of this in a few minutes. Okay, let's continue on with muscle strength. So when you're working out and you're increasing your muscle, what are you actually doing? When you actually work out and increase your muscle, right? You're actually increasing the muscle size, and you're actually increasing the fascicle arrangement. You're changing the fascicle arrangement, and you're also changing the motor unit size. So the larger the motor unit, the stronger the contraction. The more parallel, right, within a muscle, the more, or if you think about a pennate muscle, what's a pennate muscle?
Feather.
Feather shape. A pennate muscle is going to be stronger than a parallel muscle. So name for me a muscle that's kind of pennate in shape. Are you picturing the rectus femoris? If you look at it, even in the model, you'll see it comes in at an angle, right? So those are short fibers connected to the tendon very quickly, so there's a lot of force that can be generated. If it's a long, skinny muscle like the gracilis or the sartorius, not exactly a major muscle for strength, right? So your pennate muscles will be stronger than your parallel muscles. And then, of course, how big around a muscle is, you've got more myofibrils, more myofilaments. You've got more force from those increased numbers of fibers. Also, you can have that MMU, that recruitment idea. Looks like we need a new projector over there, right? But definitely you can have more recruitment when you're getting a stronger muscle. You recruit more, you get more motor units. And then this idea of temporal summation. We dealt with this in the nervous system. Tempo, right, timing. If you are sending multiple signals very, very quickly, what happens to the muscle? We saw it. That stair-step effect, right, can occur. So if you're sending signals very, very quickly, then you can effectively get a stronger muscle contraction. Also, remember wherever the muscle starts, that length relationship. If your muscle starts already in a half-contracted state, there's not going to be as much. That's true in the whole muscle, but we also saw that's true down at the sarcomere level too, isn't it? Right? So there's a distance relationship in how strong each sarcomere can contract, but that all comes back to what is the state of contraction when you begin the exercise. And then, is the muscle fatigued? Right? Is there one of those features about fatigue that has already compromised the muscle? So there are lots of things that can contribute to fatigue, a lot of things that can contribute to strength. If you want to do resistance training, weightlifting type things, then you're going to want to contract your muscle against a load that resists movement, right? You want to have a real heavy load And just a little bit at a time will significantly increase the muscle growth. Growth in a muscle is going to come from cellular enlargement. Okay. So when your biceps get bigger, you're actually, the muscle cells within the biceps are getting bigger. So what is there more of? Each cell is getting more what? Myofibrils, right? Those cylindrical structures within the muscle cell, there's more myofibrils. Those myofibrils, of course, are made up of the myofilaments, and so you get a bigger diameter of the muscle and more force that can be generated from it. If instead you want to increase your endurance training, your aerobic exercise, then you want to improve your fatigue-resistant muscles, right? I mean, some muscles are fatigue-resistant, some muscles are naturally more prone to fatigue. So if you want to be an endurance person, you want to really work on increasing your endurance or your fatigue-resistant muscles. And again, those are the fibers that have more mitochondria. Does that make sense to you? That your fatigue-resistant muscles would have more mitochondria, more ATP available. They would be more glycogen. They'd have more blood flow. They'd be red, right? They'd have more oxygen available to them. They would have a greater density of capillaries. So how does it all work? Let's finally connect again the nervous system with the muscular system. We know this. We know a lot of this. Maybe a few new words here. How do you control your skeletal muscle? How do we control our skeletal muscle? Right? We voluntarily think of it. Where do those fibers start? Where do those neurons start?
In the brain.
Where in the brain? In the
Thalamus. Hypothalamus.
motor strip. Right? We're sending out motor instructions, and that motor strip, the precentral gyrus, is found where? What lobe? Frontal lobe. Okay, so our signals are sent down from our frontal lobe. And then what? So a signal goes down, right? From our frontal lobe, down from the motor strip, down the spinal cord. And what's going to happen in the spinal cord? That neuron is going to synapse with another neuron, and that neuron is then going to leave the spinal cord and go where? To the muscle, right? Remember in the senses we talked about there was a first order, a second order, and a third order, sometimes a fourth order for hearing kind of neuron. Same idea. For you to move your muscles, there's going to be two neurons involved. We're going to have lower and upper motor neurons. So where's the lower one? From the spinal cord out to the muscle. Recall that skeletal muscle, there's one neuron, right? All the way from the spinal cord, from the anterior horn of the spinal cord, all the way to the muscle itself. We're going to see now, too, the interaction, how this is all influenced. If I said to you, are your muscles motor or sensory, or both? Do your muscles have motor neurons going to them? Do your muscles have sensory neurons coming from them? Or do they have both? They have both, okay? So we usually think of, oh, muscles are receiving motor signals. But what I'm going to remind you about in a moment is that there are also sensory neurons leaving your muscles, going back to your brain, and those neurons are going to inform your brain about your overall movement, and it's also going to help reset that perfect distance of your muscles. So there's always going to be some sensory feedback going back from your muscles and from your tendons back to your higher areas. So the muscle is not just a motor organ, it is also a sensory organ. So what do our muscles have in them that receive and send signals back in a sensory way? Golgi tendon organs, you've heard of these before. The Golgi tendon organs, those are found where? Do you remember? The Golgi tendon organs are found where the muscle and the tendon, kind of the interface of the muscle and the tendon. And the whole point of the Golgi tendon organs, it's a weird word, I know, because we think of a Golgi apparatus in the cell, but same spelling. But the Golgi tendon organs, what they're going to do is sense if there's too much stretching on the tendon. Because if the muscle is being pulled so hard that the tendon is being pulled, there's a potential for what to happen? The tendon to detach or tear away from the muscle or the bone, neither of which is good. So this is a protective mechanism in place. There's also muscle spindle apparatus. Now this one I believe is new for you. And within the muscle, there are cells that are not at all involved with contraction, but instead are reporting back about how stretched the muscle is. So when you think of muscle cells, you think of something that can contract, don't you? But there are also going to be some cells clustered within your muscles that are not at all involved with contraction, but instead are sensing stretch and are sending signals back to the nervous system. So let's talk about the spindle. If I say spindle, is any shape come to mind? I don't know that. When I say spindle, does maybe this come to mind? For some people it does, for other people it doesn't. But sort of something shaped like this, that's a horrible drawing, I know. But within your muscles, I'll show you a cartoon in a minute, there are intrafusal and extrafusal fibers. And within the muscle, there are these intrafusal fibers. They come in two types. I'm not going to worry about the difference between them. But there are these bag and chain fibers. What they are sensory cells. This is what I want you to appreciate. That these bag and chain fibers are sensory cells. And what they're going to do is report back. Okay, so don't worry about the names here. Don't worry about any of that. But just know that there are these sensory cells. And when I show you this picture here, it's going to make sense. We are looking at this entire muscle. Okay, so here's the muscle. And all we're doing right now is we're taking a look in this little spindle area. And we're zooming into this picture. The cells out here, the cells outside, the pink cells are normal old muscle cells. Okay? But within this little spindle, within this little area, here are some cells. And those are the bag and chain fibers. I'm not going to care about which one's which. But what you see is nerve, right? See the yellow nerves wrapped around? And these cells are not involved with contracting the muscle. These cells are responding or are picking up sensations of stretch. And those nerves are sensory nerves. They are going away from the muscle and are going back where? Up the spinal cord, up to the brain, right? So what we're doing is reporting to the brain about the stretch of the muscles. Now, what's that big word that starts with a P that our body knows its position in space? Remember proprioception, right?
Yep.
And that term is a big word that means that our body is aware of its position. You can close your eyes and you know that you're sitting and you know that your elbow is at 90 degrees. You just know it. You have this sense of proprioception. Why? Because some of these muscle spindle fibers are reporting back to your brain and are saying to you, oh, your muscle is, partially contracted and is at a 90-degree angle. So you're aware of these things through some of these muscle spindle fibers.
And when you improve your proprioception, is that just improving the pathways?
Awareness, yeah.
You've always have supplementing the receptors in your muscle.
You can fine-tune this a little bit. Hold on to that thought. You can fine-tune this a little bit. Let me get you a couple more terms here. So the extrafusal fibers, that means what? Outside of the fusiform. If I say the word fusiform, does that make sense to you? Have you ever heard it said that smooth muscle cells are fusiform in shape?
Yes.
And that was sort of this long, spindly kind of shape. So if I say extrafusal, it's outside of this, and the cells outside of this are what? Normal old contracting muscle fibers. It's the intrafusal cells, right? The cells inside this fusiform shaped structure that are these stretch fibers. So look what it's telling me. So the ones outside are the alpha fibers. That's just the word used. So the extrafusal fibers are the alpha fibers. They're normal muscle fibers. But there are also gamma fibers. And these are the muscle fibers inside the little fusiform area. And the contraction of those does not shorten the muscle. So they're not involved with shortening the muscle, but they are increasing the sensitivity to stretch. And they're feeding back to the neurons in the central nervous system. So again, when we think about skeletal muscle, we think, oh, okay, it's voluntary, and it's being controlled by our higher brain regions. Think what? Right? Again, frontal lobe, right? And the motor strip. But remember that our skeletal muscles can also be unconsciously contracted. Why? Yeah, and a reflex, right? A reflex is an uncontrolled movement of your normally voluntary muscles. Okay? Some protective mechanism that causes that reflex to occur. So, what's important there? Alpha fibers were which ones? Normal, regular muscle cells. Gammas were the name given to the cells inside that little area that were not causing the muscle to contract, but instead were sensory and sending signals back to the brain about sense of stretch. We know a little bit about reflexes. We're we're revisiting things we did back in Lab 3, but thinking more about them from the muscular system. So what do we remember about monosynaptic reflexes? Monosynapse means what? One synapse. For a reflex to be monosynaptic, it means that it must only have two neurons. One going in, one going out. Sensory in, motor out, therefore no what? No interneurons, right? No additional neurons inside the spinal cord. This is your knee-jerk reflex, the most simple, most, it's very, very fast, and one synapse involved. Now, the other thing that reflexes are doing, though, is that reflexes are also helping to maintain your optimal resting length of your skeletal muscles. Remember that curve I showed you? That looked like this, and it said that there was an optimal, right? And that your sarcomeres were to be 2 to 2.25 micrometers in distance. How do the muscle fibers know where to keep their resting? Because they have feedback from some of these fibers. So there's always some information. So we know about a reflex, right? We know that there's going to be a stretch. But where is this stretch? When you hit the hammer on the patellar tendon or patellar ligament, what's happening? You're stretching the muscle, but now I'm telling you that you are specifically stretching the spindle fibers. And those were the what? Which ones were those? Those were the gammas, right? And then that's going to activate the sensory neuron. And that's then going to activate the alpha neurons. Because what do the alpha neurons tell you to do? Alpha neurons are the ones that contract, right? So when you hit the hammer, you're stretching the muscle. You're stretching those intrafusal fibers. They're sending a signal that now will activate your contractile fibers and cause the stretch to be reduced on the muscle. So again, you're hitting the hammer, right? And there's going to be, let's look in it. We're hitting the hammer on the tendon, on the ligament here, but then what's happening in the muscle? The muscle just got stretched unexpectedly, didn't it? And so what happens is that you're going to activate those sensory neurons in there. Those sensory neurons are going to come in, and they're going to go directly back out through an alpha motor neuron. And that's the one that's going to cause the contraction. Now, let me back up. I just misspoke, and I'm going to confuse somebody if I don't back up really, really fast, and I apologize. Alpha and gamma here, I just misspoke. Alpha and gamma here are referring to muscle fibers. Okay? So remember the fibers inside were what? Gamma, and the ones outside were the alpha. Okay? That was referring to the muscle fibers. Over here, this is an alpha motor neuron, not an alpha muscle fiber, okay, but an alpha neuron. There's two alphas here, two different things. So we've got these intrafusal fibers, these spindle fibers, and then we also have the Golgi tendon organ. And this one you know more about. These are constantly monitoring the tension on your tendons. And they're sensory, right? They're picking up sensory information. And if, again, the tendon starts getting stretched, then it will cause the muscle to do what? If the Golgi tendon organ is activated, it's going to send a signal back to the brain that tells the muscle to do what? Contract or relax? Hold on, let's back up. When we strike the patellar ligament and we stretch the muscle and we activate the spindle fibers, the muscle then did what? Contracted, right? So we contract in the reflex is to contract. But if I am stretching my muscle and the Golgi tendon organ, which is at the muscle tendon border, is activated, what is it telling my brain? That the muscle is being stretched too much. So what is the brain going to tell the muscle to do involuntarily?
Relax.
Relax. Because to contract more would to create more tension, which would go further to damaging the tendon organ, right? So if you've ever watched a bodybuilder or some Olympic kind of sport, and they're going to go for the guy from Poland going for 800 pounds, right, or whatever it is, right? And he's going for this massive lift, and all of a sudden his body looks like it goes to spaghetti. What's happening? His body just said to him, hey, dummy, right? You just activated your Golgi tendon organs to the point where your body said, relax those muscles. Involuntarily, they drop, right? If you watch anyone going for their maximum weight, now not the weight they're normally doing, right? The weight they're normally doing, they built up to a point where their body is used to it. They've built up their tendons, they've built up their muscle. But in a competition, where now it's the last three guys, and now you're going for something you've never done, but you want the gold medal. And you're going for that top medal, that top weight, right? And you're going beyond where you've ever been before, at least not safely. And you'll see these guys just go like a limp, like a noodle, right? When they're going for that lift. Those muscles just relax. You might think, oh, they gave up. Oh, no. They didn't even try, right? Their body said, no way, buddy. And they just drop it, right? Because their Golgi tendon organ said what? Not this time. So here in the cartoon is the Golgi tendon organ. If we start to stretch this too much, and this is activated, it is going to send a sensory neuron to the spinal cord. But what it's going to do is send an inhibitory negative signal. And that motor neuron is going to be inhibited. And what's that going to do? Not send the signal to contract. So in essence, it's going to cause the muscle to relax. So we got two different protections here, don't we? Right? Spindle fibers against being stretched unexpectedly, causing the muscle to extend, right, or to contract, and the Golgi tendon organs causing the muscle to reflexively relax. Two different sides of the possible situations. So in the knee-jerk reflex, right? Now, I always say to you, oh, in the knee-jerk reflex, it's monosynaptic, right? Mm-hmm. But wait a second, is it really that simple?
No.
Because not only, when I hit the patellar ligament, not only am I kicking out, okay, so I flex these, but what did I do to my hamstrings? I relaxed them. So those instructions had to come from somewhere. right? So even though, yes, the knee-jerk reflex is monosynaptic, and that's true to the quadriceps femoris muscles, there are still other interneurons which are going to be stimulated, which are going to go to the antagonistic muscles, right, and inhibit them, allowing for those hamstring muscles to be extended. And it gets really complicated. I mean, there can even be reflexes that go to the contralateral limb, right? If you step on a tack with your right foot, not only are you going to lift your right foot, but you're also going to extend your left foot to hobble your weight on it. So again, these reflexes are very, very complex. They're simple in one way, but they're also quite complex in the way that they help us. So for example here, if you look at this one, you've got all this going on. Not only when I hit the patellar tendon or patellar ligament and I cause a gastrocnemius, I'm also going to inhibit signals going to my hamstrings. Okay, that has to all happen simultaneously or else we wouldn't be able to do the job. And then this is just showing what happens in that contralateral relationship. So let's say I step on something sharp, right, with this foot. So those sensory signals are going to come up and they're going to tell muscles on one side of the body to flex, the antagonistic muscles to extend, and then it's going to go to the other side and also inform the other side, right, to do the opposite, if you will. So you're going to lift up your right leg because you just hurt yourself, but you're going to extend the left leg. Well, what's involved with extending your leg? I mean, you've got a lot of muscle groups, some of which are contracting and some of which are having to be relaxed to do that job. So I mentioned it, but I never showed you a slide on this. Upper and lower motor neurons. There's two neurons involved with the control of your muscles. The upper neurons are the ones that leave from your pre-central gyrus. You know this as your motor strip, right, in your frontal lobe. There are also some upper motor neurons originating in your cerebellum. It's involuntary, but clearly you still have signals going down from your cerebellum, down your spinal cord, informing your overall muscle coordination. And we don't want to forget about the basal nuclei. Remember, when you hear the word basal nucleus, you think an area of motor control. So this is an area of the brain that's also sending signals down your spinal cord that are helping to work with your overall muscle work. The cerebellum also, remember, is working with your vestibular system, right, and your basal nuclei. I haven't talked about the red nucleus this semester, I don't believe. So don't worry about the rubrospinal and the red on there. But everything else you can appreciate. So that means that when you're moving your muscles, right, there are upper motor neurons that are originating from your motor strip, from your cerebellum, or from your basal nucleus. And those signals are going down to your spinal cord. And there they're synapsing with lower motor neurons. And those are the neurons that are going out to the actual muscle, right? Upper and lower. When we were talking about senses, we talked about first order, second order, and third order neurons. They went up the spinal cord, didn't they? Now we're talking about motor neurons. They're descending fibers, and they are going out to the effector. Now, Ty, I see your book open, which is perfect. The next slides were some of the ones that I told you I was going to skip over. Okay? And if you will write down this page, these next slides, everything I'm saying in these next few slides are coming from page 431 to 435. It's only 4 pages, right? So the last 4 pages of chapter 11 deal with cardiac muscle and smooth muscle. And when you're reading through this, you can read through the PowerPoints or you can read through your book, but I want you to realize this is only 4 pages of reading. It's not going to kill you. And as you're going through these 4 pages, what you're looking for are, again, those comparing and contrasting ideas. How are you going to learn that cardiac muscle is different from skeletal or different from smooth? Now, we're going to see these again. When we get to the cardiovascular system, I'll be talking about cardiac muscle a lot. When I get to the digestive system, I'll be talking about the features about the smooth muscle. And when I get to the vessels, I'll also be talking about smooth muscle. So you're going to hear it again. But I want you, these slides have the little red star on them in the upper left-hand corner, and these were the slides that I told you you would be doing on your own. Now, I want to spend 2 minutes just flipping through them, but I want you to realize I'm not going to go through these in detail, and you can pick up on these ideas. So cardiac muscle, found only in the heart, we know this. It looks different. What's the one thing you're looking for? Those good old intercalated discs. Obviously, they're very resistant to fatigue, and for that, we're thankful. Right? These cells just keep on going and going and going. Striated, right? No doubt. You know this. Cardiac muscle is striated. The cells themselves are shorter. They're called cardiocytes. Oftentimes, cardiocytes, the cells of the heart. And of course, we see those intercalated discs. There is a less developed sarcoplasmic reticulum, but there's still plenty of calcium in the area, okay? So we still have some sarcoplasmic reticulum. We still have, they're striated, so we still have actin and myosin, right? That's still very much the same in cardiac muscle. One of the differences, though, if you damage your skeletal muscle, you have satellite cells there that are stem cells that can regenerate your muscle to some extent, whereas the cardiac muscles do not have that. So if there's damage to cardiac muscle, you instead get fibrosis. You get basically a buildup of scar tissue. So a damaged heart does not restore itself, unlike a damaged skeletal muscle. So that's a big difference, right, between skeletal and cardiac muscle. The cardiac muscle is autorhythmic. That is, there's a pacemaker. We'll talk a lot about that in the next chapter. Within the heart, there's a part of the heart that actually sets the rhythm of the heart. More about that later. And the heart uses aerobic respiration almost exclusively. It has a lot of myoglobin. It has a lot of glycogen. And the heart cells have an unusually large mitochondria. So they are just ATP producing factories, these cardiac myocells. Okay? So much, much bigger. And then we get into a quick conversation about smooth muscle, and I'll let you read through those last few slides together. So there's a few slides. Again, look for the differences between them. And then it finishes up with muscular dystrophy. We've already talked about muscular dystrophy. And then lastly, a slide on myasthenia gravis, which you can read about as well. And that finishes up this chapter on muscle. We'll be starting the cardiovascular portion of this next week. And again, check out those last couple of pages. It might be good just to read those last few pages about cardiac, smooth, and skeletal muscle and how they are different. That finishes up our week, folks, so enjoy.